This presentation describes the growth of mycoplasmas and other synthetic derivatives used in a lab on agar medium in cases where you need to select organisms or to titrate their concentrations. One of the issues with mycoplasma medium are that they are very complex. They contain proteinaceous serum or other components, and also other heat labile components, and therefore they have to be treated in slightly different ways. Furthermore, that the agar media using these complex medium have to be treated physically in ways that are different from most bacteria. In the lab at JCVI, we use the SP4 medium, and it's stored in two parts. The part one is the either liquid or with agar, which I'll discuss today. These can be autoclaved and stored at room temperature. The other part two contains the more labile components. Those are made in liquid, sterile-filtered. So we need to combine part one and part two, whatever form of part one we have, in order to make a complete medium. When making the agar medium, which I'll be discussing today, we have to do that at temperatures that allow it to eventually solidify, but not to create any problems with the labile components. The agar surface that we form has characteristics that are slightly different. With these wall-less mycoplasmas, they actually grow into the agar. They'll be growing down into the agar matrix, so the agar concentration is lower. Also, the agar surface is easily disturbed and is denatured by drying out or by certain types of heating procedures, which I'll talk about in a minute. So we'll be discussing the preparing the solid medium and pouring agar plates, as well as how to deliver the organisms to the surface of that agar. The video starts showing the solid agar medium in part one, which can be microwaved to boiling multiple times to get it completely dissolved. And then that solution, part one solution, can be equilibrated to 50 degrees and maintained prior to this procedure. The other part two, could be quickly warmed to 37 degrees and kept at that temperature for the procedure. Under sterile conditions, the two components will be added. The volume ratios in this case of part one to part two are 3:2. You simply add those into a single bottle and mix by swirling. Of course, this will create bubbles. You want to avoid bubbles throughout the whole procedure, but by this sort of gentle swirling, you can minimize that. People can pour from these, but there's a tendency to get enormous numbers of bubbles. To avoid that, we deliver through a pipette. These are 100 by 13 millimeter Petri dishes, and that takes about 25 mLs of the agar medium. To do that, we sort of either pre-warm the pipette or bring fluid in and out of it a few times to warm it, and then using a 25 ml pipette and loading it to about 26 mLs or so. You can very carefully put the tip without the bubbles in the tip if possible, and gently deliver this to the Petri dish. We do not expunge all the fluid. We leave fluid in the pipette to avoid the many bubbles at the meniscus in the pipette. We'll talk about bubble removal in a minute, but this can be repeated into as many plates as you wish, working fairly quickly to avoid the fluid setting up in the stock bottle. Again, ejecting the fluid, but not ejecting all the bubbles at the very end. Bubbles should be removed or can be removed by letting them essentially float into the tip of the pipette. And we'll see a few cases of this. And the reason for this is to avoid other methodologies for getting rid of bubbles. In other cases, with various plates, people will actually use a flame to break bubbles. This is absolutely not good for these plates because they contain proteinaceous materials, and this process essentially sears the surface and makes them unusable. So bubble removal is always important, but this is one method shown here that allows it to be done without any kind of a denaturing process. These plates should be poured, of course, on a flat surface so that the depth is uniform around the petri dish. They will essentially set up in about 5 minutes in normal at room temperature, and they will be cooling, and you'll see fluid condensing on the lids as they cool. Getting rid of that fluid is important and can be done by a number of methods. It's important to dry these plates by airing them out under the laminar flow or leaving them open for about probably 10 to 15 minutes to reduce the amount of moisture in the plate. These will be stored inverted, and they will condense further during storage. One way to avoid the bubbles or the drops in the lids is to, while they're still inverted, taking them out of the 4-degree environment, removing the lids while inverted and putting a new lid on is one way to avoid contamination of the dripping fluid from the stored lids. Again, these can be put into a plastic sleeve, and stored inverted at 4 degrees for further use. In part 2, we'll show simply the way to deliver organisms to the agar surface without disturbing that surface, and in a manner that will allow you to look at colonies in 2 or 3 different ways. We'll either be delivering very small volumes, so multiple small volumes in a pattern for such things as getting a rough titration by colony-forming units, and then quickly we'll show you the delivery of larger volumes, which you might use for selecting colonies under selection under a antibiotic marker. In the first case, we'll look at small volumes where we've made dilutions of organisms for a titration, using delivery of very small volumes to multiple spots in a single plate. So the video starts showing the delivery of about 10 microliters in this case to the surface into a small pool. Again, not touching with any physical object, so the liquid is dripped onto a small pool, if you will. This can be done repeatedly with multiple small deliveries, spaced so that they do not run into each other. And this can be done multiple times around a plate, as you'll see in one case. It's again good not to puncture the surface with the pipette. These are allowed to just dry, and you can see they will sit there and not mix, but they will eventually soak in and dry, at which point the plates can be inverted and stored at 37. The other thing to show is the result of the titration, where you have colonies formed at from the different dilutions. In this case, 2 different organisms. You can see where at high dilutions you can achieve separated colonies where those can be actually picked and used, or the whole thing can be used for approximate titration in colony-forming units. In the next frames, we'll show the delivery of a larger volume for the purpose of perhaps selecting antibiotic-resistant colonies out of large populations. This is about 200 microliters, and the idea here is to avoid getting it to the edges, but spread it by this orbital rotation to deliver it to as wide an area as possible. You can do this in different ways, but the thing is to avoid a physical spreader, which is used for some other types of organisms. These are then dried and used, incubated for the period of time required, and then colonies can be observed under the stereo microscope.